Hi, today I'll be talking about prokaryotic transcriptional regulation. An operon is a cluster of structural genes and transcription controls. It is not present in eukaryotes and has a promoter, an operator, and structural genes. RNA polymerase binds to the activator binding site on the promoter, which is a part of the lagging strand of DNA. The operator is connected to the promoter and is the binding site for the regulatory protein. There are 4 types of prokaryotic transcriptional regulation, but I will be focusing on negative inducible regulation. Negative control means there is a repressor regulatory protein present that binds to the DNA to prevent transcription. Inducible control is when transcription is turned off, but is turned on when there is a small effector molecule present. To further explain the negative inducible regulation, we will dive into an analogy. Imagine a classroom and the students are talking a lot, and the noise level represents transcription. For transcription to occur, the RNA polymerase has to attach to the promoter. In this scenario, the RNA polymerase is the sign on the wall that says speaking is allowed. The teacher, Ms. Wall, is trying to bring the noise level down to 0, and in doing so, acts as a regulatory repressor protein. The repressor protein can flip the sign from on to off, causing transcription to stop. However, another teacher from down the hall, Mr. Stiles, comes down to talk to Ms. Wall about a staff meeting. Ms. Wall gets distracted from trying to bring the noise level down and converses with Mr. Stiles instead. The sign on the wall stays at on, and the RNA polymerase allows the transcription to occur and the noise level to stay high. Mr. Stiles is playing a similar role to a small effector molecule that is an inducer and an operon. In the operon of the classroom, transcription is on until Ms. Wall represses the noise level. However, Mr. Stiles, our inducer, distracts the repressor away from the operator, and transcription can occur. The RNA polymerase stays attached to the promoter and the sign stays on. So how exactly is transcription impacted when there is and isn't a small molecule, the effector, present? When the effector is present, transcription can continue because it binds to the repressor protein and prevents the repressor protein from stopping the RNA polymerase. When an effector is not present, transcription stops because the repressor protein can bind to the operator and stop RNA polymerase from transcribing the DNA. Ms. Wall is the repressor, and without Mr. Stiles, she would have stopped the noise levels and transcription would be off. When Mr. Stiles came to the classroom, Ms. Wall did not flip the sign and transcription continued. I hope this video helps you understand negative inducible transcription regulation. These are my references. Thank you.